speaker. One is on the Analytical with Hanspeter, Michel, and the other one about the NMR by David Fry. Hanspeter is a scientist at Hoffman La Roche. I know him now for almost 20 years. I will tell you a secret. Some days I love him to death, but some days I hate him. I wish I kill him because I come and I give him my peptide and my thinking is over 95% pure. He looked at it and said, "What give me for a Dreck?" Dreck, that's a German word for schmutz, or what is an impure peptide. And he looked at all small peaks here and said, "Look at this." I said, "What?" "I don't see them." He sees them. And then he, being analytical and mass spec chemist, we fight about one unit mass spec. So what all the fight? 1 unit. He usually wins at the end. So please, Hanspeter, tell them what I did wrong.
Oh, you didn't do anything wrong. I think it's okay. I can be heard. So CMC is the problem. Yeah. It's not scientists at CMC that's it's the cause of lots of issues. I'm working in a formulation department with Roche. But within that department, I'm representing the analytical activities, of course, along with many other colleagues. I left the abstract there, not to read it and not to talk about it, maybe just for myself, to sometimes keep myself reminding what I wrote, what I would be presenting. It does look like a small print statement, which a CFO would put up first, and talking to the shareholders with forward-looking statements. So there are no forward-looking statements here. They're all backward-looking. Peptide analytical toolbox. We always need to put things in a simple form, so to make it easy to see what we have to do in a development process. So I differentiate between the peptide API characterization and then the peptide as a drug product. I might have forgotten or omitted some additional methods, but I believe that these are the major tools we're dealing with. Certainly HPLC, UPLC is one of the staple works, ID testing, purity, stability, and content, and not far behind, of course, MS and LCMS, which also the ID testing, purity, stability, structural characterization, sequencing. I'm talking about this ID testing, purity, stability, and these things because if you go forward in development ahead from preclinical into clinical, that's what we have to do, all these activities. Amino acid analysis. We still do amino acid analysis. In our case, mostly for quantitative, for example, reference standard characterization. It has some niche in the epimer characterization. We do Edmond sequencing for obviously structural characterization of not only peptides but proteins. We also have mols available, or size exclusion mols for oligomer and aggregate characterization. Oops, that's not the right button. We do not NMR in our lab, but we have NMR experts here. So if there's anything NMR, I will talk to Dr. Dave Fry, and he will help us out. And then, last but not least, infrared and Raman infrared can be used for ID testing. The peptide drug product characterization. Essentially, these methods are the same on the top. There's not much changed what would be done with all these methods for the primary sequence analysis, primary structural analysis. Only with the drug product, we getting a little bit, we have to go a little bit further ahead in, for example, particle size characterization, zeta potential, mobility, all these things, moisture content, characterization of lyophilized products. Of course, we have to worry about sterility and endotoxin, colloidal stability, viscosity, and syringeability. So some of these things are actually quite important for further down the line. I think one of the very important things is viscosity. We always sort of neglect at the preclinical stage in the discovery phase that we have to later formulate these things, and we have to formulate it at the right concentration and in the right amount. And not knowing the efficacious doses, you can put a lot of trouble in that area if one doesn't take care of that very early. I had this sort of represents the same listing of methods, just a little bit in a different way of presenting it. With all the method development and what happened in the past with Edmund, amino acid analysis, and particular mass spectrometry, and of course all the other methods, we have methods available for the primary structure analysis. I have put that in green because we always try to say how much effort do we have to put into developing a peptide or a product. Green means that's what our management likes, green. So we have the tools, they're available, there's no unknown, they're not necessarily easy, but there will be no problem with along the line. Now, we go a little bit the higher order structures. So these are aggregation, gelation, we have to deal with solubility, we have to ask about conformation, where we have methods like CD, NMR, infrared, or also MS for that matter, or even the topic about the immunogenicity potential. That might even have to move over here, because I'm not sure whether that is. Yellow is sort of intermediate, where we sort of think we know what we're doing, but sometimes not. And then we go into the area of a complex drug product, and I don't want to spend too much time. I might come back to that a little bit later at the end of the talk, but it can get quite complex and quite extensive. And with that, of course, we always have to talk about resource needs, and so that's where we're flowing from the green area into the red. And we don't want to see red. We really want to turn that all into green. But the field goes forward with all these things. It's just it takes time to do that. The first topic I want to touch upon for peptide characterization, mass spectrometry. That is I have 2 slides about that, and that is a very, very shabby and really minimal slide to describe mass spectrometry. There are thousands of publications, and it's really enormous. I could talk for I don't know how long, forever, probably, about just that topic. But I had the fortune, I was lucky, I have been in mass spectrometry of peptide for 25 years. So I have seen the very beginning to what's happening now. And the very beginning, I was lucky, I was able to join Professor Don Hunt's lab in Charlottesville. So when I first started, or I should say we, because it's not just me, but in that particular case, when I tried to sequence my first peptide, we used for one mass spectrum, we needed about 100 picomoles of material. And I got, we had an old Finnegan instrument, I got the hard drive put into my hands. The hard drive was almost the size of a Swiss cheese. Not quite that heavy, but I mean, that was a really big thing. And it accommodated about 100 megabytes of data points. So that was really very early. And in order to achieve the sensitivity with 100 picomoles, we had to open the resolution plus minus 2 to 3 Daltons to get enough ions. Now, if you do that plus 2 or 3 Daltons, you can solve puzzles, you can put sequences in there, but there's always the ambiguity involved with it. So we did a lot of chemistry and all that to figure out sequences. in that thing. But when I started with a mass spectrum, to achieve a mass spectrum of plus minus 2 Dalton from 100 picomoles, after 4 years when I left the lab, I have to mention that was before electrospray and before MALDI. When I left the same lab 4 years afterward, electrospray and MALDI were introduced into field. And when I left, we acquired the same mass spectra with 100 femtomoles. So for 4 years, order of magnitude of at least 1,000. And it just exploded into the mass spectrometry of peptide as the field exploded. Nowadays, we have all these beautiful instruments, very wide application range. You can use them for almost anything. High analysis throughput, enormous high sensitivity. My colleague Dr. Dave Fry can tell me something about the sensitivity, the discrepancy between NMR and mass spec, so that's another topic. There are little cons also to it. It does still require, also some instruments are really very easy to do. They don't need major expertise, but so some of the more complicated experiments, they do need some expertise, and they are a high instrument cost. So I don't want to call myself a mass spectrometrist because my education is biochemistry, but my heart belongs to mass spectrometry. And my Christmas wish list is here. And I'm always told that's not possible. That's too big a list. So we have to pick out the best out of that. And this mass spectrometer, I believe mass spectrometry companies, they're also very skilled to make sure that we don't have just one instrument which can do everything. I don't know whether it would be it's not possible from how the technology works, but they really try also to sell all these things. And our management sometimes doesn't understand that we need all these different things in the same place to do what we are doing. So in particular, Orbitraps, they're phenomenal. I'm lucky I have an Orbitrap in our lab, and it's really excellent instrument. It sort of does replace FDMS to a certain degree because of the accuracy and the resolution. We do acquire mass spectra, and if molecules are not too large, we can extract elemental composition from the mass accuracy, which is a tremendous tool in the characterization of impurities and degradation products. We do run quadrupole instrument, triple and single quadrupole. We use triple quadrupole instruments for the quantitation. There's nothing as good yet as a triple quadrupole instrument if we want to quantitate something. So none of the other instruments ever reached that powerful possibility. And we have all the series of time-of-flight instrument, time-of-flight QTOF, QTRAP is also can also be integrated in the quadrupole. They are beautiful instruments for very high speed analysis, and I need to be careful with that. Very high throughput analysis, but also very high mass range. This is incredible what the field in this 25 years brought. Not only peptide, when we started 25 years ago, was maybe 1,000 Dalton or 1,500 Daltons. Nowadays, with time-of-flight instrumentation and the proper ionization, people are looking at whole mega protein complexes. So they're spraying enormous, huge things into this mass spectrometer and get molecular weight information from it, which is incredible. It's mind-boggling. And recently also, ion mobility time of flight, which sort of does add an additional dimension to the possibility of separate molecules. Ion mobility allows you to differentiate isobaric molecules based on their conformation and based on their structure. So if Walid said we give him a headache with our instrumentation, we don't have ion mobility, he would be surprised how much more a headache we could give him if we add ion mobility to the analysis. But it does almost replace the need for other primary structural analytical tools. I will talk a little bit about these other tools. Very short, just very brief. Mass spectrometry for of course, the primary structure analysis, including sequencing, quantification, purity assessment, impurity degradation product characterization, non-covalent interactions, and also a very fascinating topic which can be done with laser multi and mass spec is tissue imaging. So you can actually map peptides, small molecules and proteins within tissues. Unfortunately, you cannot go into the cellular level, but as far as the tissue imaging is concerned, very powerful tool and a fascinating development going on there. Now, the other tools, element sequencing there, N-terminal element sequencing, and not very many laboratories would still do that. It really did lose its luster. It has some advantages. It is a very simple data interpretation and relatively simple sample preparation. For some protein discovery protein analytics, it does offer some really good applications to get sequence information easy and simple. It has a niche in the peptide analytical world. It's a rather narrow application range. It's not cheap in itself because aside of manpower, it needs the cost of the chemicals and it's a slow throughput. Also with that slow throughput, that's another story. When I was in Professor Hunt's lab, that was the starting of the peptide sequencing, and we actually did have an Edmund sequencer running parallel to the mass spectrometer. And I always was thought we tried to figure out mass spec should be much faster and much higher throughput. At that time, because to actually achieve a sequencing, a 100% correct sequencing readout, we had to do a lot of mass spectrometry, a lot of chemistry, and sometimes we even had to synthesize the peptide and reconfirm that our structural assignment is correct. So I sometimes said, I'm in a hurry, I'm putting it on the sequencer. That doesn't work like that anymore. We use it for reference standard characterization. The readout is very unambiguous, very simple. We use it for impurity characterization. Sometimes we have strange impurities, we don't know what it is. And one other very good application for this instrumentation is the determination of PEG modification sites. There's a good reference available. It's a review article which more or less describes the history and the current application if somebody is interested to do some more reading. Now it's getting even worse. Walid told me it's an advantage in peptide analytics. So what we're doing with the amino acid analysis is 50 years old. And we actually still do the method which was originally invented by Stein and Moore. Generally, you have different hydrolysis modes, you have different readout, including mass spectrometry. Why we're doing it, it's here, that's the quantitative readout, the characterization of reference standards, the definition of label claims, and bringing projects forward to the clinic. We're really dependent on that quantitative tool because it is a direct analytical method for determination of peptide content. And specifically recently where peptides are not just, they don't seem to be just peptides anymore. They are polymer, they're bound to polymers, they come in PEGs, and they call in all sorts of shapes. It's a very efficient method to obtain this quantitative readout, and we do obtain reference standard amino acids from Fluka and Sigma, which does include certificate of analysis. If we go forward, our quality control units, our quality assurance units, they like certificate of analysis. We can trace back our quantitative readout to existing certificate of analysis, then we're doing fine. 2 references, if somebody's interested to read a little bit more about it, are cited below. Now, I would like to switch from a little bit general approach to a case example, and I don't want that to be too much focused on as this peptide, because I would like that more as an example of how what we have to do to bring something like this forward, so that the methods are a little bit specific for that case, but generally they're applicable for many other forms of peptides bound to polymers or liposomes or wherever they could occur. That's the peptides, 15 amino acids, plus a PEG. It's a 30 kilodalton PEG. I will say a little bit more about the structure and the characteristics of this. I just want as an introduction, a flow of thought to have that shown. I'm going to talk a little bit about the analytical role in the definition of or the discovery of the clinical candidate we had, and then within the analytical challenges, summary of analytical methods, some thoughts of impurity determination, maybe go back at peptide content, structure, oxidation, and some talk a little bit about if time still is okay, stability indicating methods. The peptide, what we got at the beginning, we decided we wanted to pegylate it. It had 2 pegylation sites. The 2 pegylation sites, the peptide that we started with, were one was in a free N-terminal, free N-terminus, plus a lysine in position 2. And when we initially started to pegylate that, we did, of course, our readout at the time was food intake. With the biologists, they measured food intake in mice. We pegylated it. We found good in vivo efficacy. We also had 2 positional isomers, and we had a very abundant dipeg species. So we didn't like the dipeg because that's troubles for purification. We decided to remove the lysine in position 2 and then pegylate the N-terminus. We got obviously no dipeg, but also inferior in vivo efficacy. So this was no good. So we started to purify. We went back to this, improved purification. We made sure we had less dipeg. We still had good in vivo efficacy, but we didn't like the process. We didn't really like how it looked, and so we had to go back. And then, of course, that's all with the help of Dr. Walid Danhau. He knows that better than I do, don't you? So he put he left the FMOC group on the N-terminal end. We pegylated it to achieve only monopegylation, and then after the pegylation, removed the FMOC to have a free N-terminus, which gave us good in vivo efficacy, no dipeg peptide. We run into another problem there. We saw a little bit smearing out of our, that's reverse phase. We saw the same thing happening in sizing, only it was sort of smearing out towards the later elution time. So we had to talk to the chemist who synthesized our PEG reagent. And of course, afterwards, we were always smarter, and it's always easy afterwards to say, but we really, we sort of at that time, with regard to that example, we were at a hard time that peroxides and PEGs are really no good. And you have, in a pegylation, whenever you do synthesis and whatever you do, you have to really watch out. You don't have any latent peroxides in the PEG. If you have just small amounts there and you don't see it, it's difficult to analytically determine. But when you put these things on stability, then it will cause troubles. We ended up in that candidate. We had no dipeg. We had a stable PEG. We had good in vivo efficacy. And also along the line, we switched from some of the chemistries we did, which are not free available PEG reagents. We did choose a PEG reagent which was of public knowledge, knowing that the cost of goods would be an issue. Now, I did forget one thing I wanted to mention at the beginning, so I need to put that back into perspective. We did develop the product for first. It was started as an anti-obesity drug, and then when it showed promise that it would work for type 2 diabetes, of course, it was channeled into that franchise as a type 2 diabetes medication. And that's why the question of cost of goods, we heard quite a bit yesterday also about that. It's quite important to keep that in control. That shows a little bit in detail the synthesis. The FMOC protects the N-terminal end to be pegylated, so we have the lysine here at the pegylation site. The PEG-SSA activated reagent in the presence of boric acid in the basic, and then after the pegylation we use piperidine to remove the FMOC, which leaves us with that molecule which then has a free N-terminus and has the PEG attached to this lysine in position 2. So sorry for that. I just want to make sure that I'm going to not lose my thread. We had to come up with analytical methods because it's a pegylated peptide, and as you will see later, the characterization of pegylated peptides is not straightforward. It does have some challenges involved, but it's jumpy. We need to worry about an identity. Normally use reverse phase HPLC. We can use peptide mapping, MALDI-TOF MS, LCMS. We have to worry about high molecular weight aggregates, impurities and degradation products, reverse phase HPLC, size exclusion chromatography. And one of the topics I will address later on, so we have chromatographically not separated impurities. We have to worry about free PEG and do that. So that shows the structure, the entirety of the molecule, and it's also summarized to a degree what kind of methods we used. So we tried to access structural information, and not only for the molecule but impurities to cover the whole molecule. In order to do that, we really had to integrate all these different methods, the MALDI-TOF for the the molecular weight of the total API. We use triptych maps. We get access to the C-terminal portions of the peptide. We use electrospray MSMS, and we get access to this part of the molecule. It turned out it was quite important in that particular case to use that because of that unnatural amino acid there. And we use Edmond sequencing to have access to the identity of the first amino acid at the N-terminal end. Our MALDI looks like that. And when we do ID testing, when we do electrospray, I will go back to this a little bit later to talk a little bit more in detail about that. That's how it looks. Now this, more or less, this looks still the same, but yesterday, Volker Schellenberger from Exton, he showed a real mass spectrum of a PEG about that molecular weight. So technology, when we acquired, did make some advances. So that can be now resolved into the individual PEG species. And it's not just, a colleague of mine sometimes called multi-TOF MS chromatography, not really MS. So at that event, what we have seen yesterday, we are also for PEGs in mass spectrometry and not necessarily in chromatography. But multi-TOF confirms molecular weight, and electrospray MS confirms the peptide part at the C-terminal end. I would like to add, we didn't use these methods at the time when we tried to develop the molecule. Multi-angle laser scattering is light scattering is very helpful. Wyatt offers that instrumentation together with the excellent course. I do like now moles. I never liked it. The reason why I didn't like it is because I always thought determining molecular weight is only possible with mass spectrometry. And the colleagues from Wyatt actually convinces us that it can be used as an absolute means to also determine molecular weights. And not quite as accurate, not quite at the resolution, but it's very helpful in polymer analysis and polymer peptide conjugates and these kind of things. And another development I found, as from the paper 2009 found, I think these colleagues from Eli Lilly, they found that the addition of amines into the PEG product analysis for LCMS allows them to shift the molecular weights to higher molecular weights, and then in combination with time of flight instrument readout, they were actually able to resolve in individual PEG species up to 40 or 60 kilodalton. So that's really different now compared to the pure multi-TOF. Now this is, that's how it looks. We talk a lot about the PEG-related peptides, and then most of the time people put the little peptide here. Most of the time the peptide is bigger. And then on the side there is a PEG or what was that? The colleague on Wednesday afternoon, was that Alenix? What's the company's name? CEM. You have to help my memory, but the definition of CEM, a clearance, what did he call it? Anyway, extension of lifetime, a clearance inhibiting moiety. So he showed a PEG and then a red moiety. And I like that, the red moiety, but the red moiety, I don't know in their case what it actually represents, that red moiety, but sometimes it really, these moieties we attach to the peptide to extend the lifetime, that they're not quite as well defined as they should. They're actually, it can be quite a nightmare. And this PEG is, of course, it's not just this one here, that's just one example with this chain length, but there are hundreds, maybe 100 or more different species in there, which constitute the polydispersity. And that sort of summarizes the challenge coming from the slide from before, which we start with the FMOC protected peptide. In this case, it turned out we had it at 87.5% purity. That was not from Wally. That was we got that from somewhere else. So no, it's dependent. Somewhere better. And then we use a PEG reagent of 83% purity, and then the chemists, well, they come up with this one here. It's really difficult to purify these things. So they say, yay, we have 97% purity, no problem. That's going to. They also, not only Walid was sometimes upset about me, but the chemists didn't like me because I told them, no. That stuff is still in there, so you don't have 97%. And so so we had to summarize sort of the type of impurities which could occur in there. I mentioned something a little bit that you had to deal with, the dimerized forms, peptide-related impurities, dimerized forms, forms with higher aggregation, and non-pegylated form. And then what the challenge actually turns around, what the challenge turned out to be was the question around the monopegylated impurities, because all the impurities in the starting peptide, they're pegylated and they're going to be carried over in the final API. There was a question, of course, again about the pegylated impurities. So from our 88% purity, a peptide, when we went forward from TLP into GMP, that would actually be an interesting story also about the interaction of how peptide analysis, how we could actually interact. They were from the same as a CRO, from the same manufacturer. We could actually feed back that information. We met with them, and from that, our batch for the going into the first GMP for the phase 1, drastic improvement, and that's ideal, and that's really what one likes. Maybe not quite. Some of these were in the 2-3% range impurities, but we want a product which covers lots of impurities, and then we want to get cleaner in going into GMP. I show that list here, not to go with details to it, but since we do have this problem about the monopegylated impurities and the difficulty to separate these things, we were very adamant to have reasonably good analytics on the starting peptide, including specifications and release testing on the starting peptide, assuming that peptide impurities would be monopegylated, but wouldn't really increase in the abundance. So at a certain point, we took some of the impurity numbers from the starting peptide to define later our purity of the product. Now, this is the molecule. It shows it a little bit. I can go a little bit more in detail. How much time do I have? 5 minutes? Yeah. Jesus. It's impossible. Are you sure?
It's a Swiss watch, yeah.
I didn't even know half of bad timing. I talked too much about the beginning. But anyway, that's the peptide originates from the lab of Dr. Walid Tanhau and Dave Fry. Dr. Dave Fry had his doing too. So that's what they came up with. A beautiful peptide. It's a derivative of PYY, shorter. It has only 15 amino acids. It has one unnatural amino acid. And it was efficacious. It's manufacturable. It's small molecular weight. Everything, no problem with it. Now, I'm in the formulation department, and I'm the analyst. The biologist, the chemist are excited about it. As an analyst, I'm also excited about it. But as a formalist and worry about shelf life, how the heck can you put that many problems in such a small peptide? So we went back to Walid, and we told him, you have to get rid of the PQA, you have to get rid of that asparagine, the tryptophan, we didn't like that neither, and so on, you know where that's going. So It's not that bad. I mean, it's all relative. That's the fight between people who want long shelf life and people who want to have high biological activity. Somewhere along the line, it doesn't look very different from for almost all the other peptides we were dealing with. I think I'm just going to maybe just talk about one topic as far as the impurity characterization is concerned. There was a unnatural amino acid, PQA, there, and we did actually observe in the starting peptide higher abundance of oxidized PQA. And we were sort of worried about that. How do we characterize that? How do we control that? How do we make sure that instability that wouldn't increase? And then so that's the task was to try to quantitate this oxidized PQA here. The basis for that is in the fragmentation. Now we're fortunate because the peptide, the PEG, is somewhere there attached on the lysine, right in front of the pKa. And the peptide, when we add a little bit of energy, we don't need to add too much energy from the mass spectrometry, it likes to fragment right there. And when it fragments like right there, it gives this discrete molecular weight there. That represents the C-terminal remnant of the peptide. If we don't add the energy, that's just a mess. That cannot be deconvoluted. It doesn't have any information. But we can spin that further. We can create that peptide in the mass spectrometer, and then we can further fragment that created peptide fragment. And we further fragment that, we get the sequence information along that peptide. And along the sequence information, we get also the first ion on the N-terminal site. We get an ion at 271, which represents the PQA. And that's the basis how we can actually quantitate PQA in the pegylated API. We can ask the mass spectrometer, represent that. We can do a parent scan and ask the mass spectrometer, give me all molecular weights which would give us a fragment of 287. 287 is, of course, the 271, which the PQA would be, plus the 16 for the oxygen. So we tune our third quadrupole on 287, and what we're getting is a signal, which is that one there. If we compare that with our GMP lot, we don't see that. And so that's basically the difference between the two lots, and it really is go parallel with what we observe in the peptide. This kind of approach allows you to ask other fragmentation. You can ask for adducts, you can ask for degradation products, you can choose what molecular weight shift you want, and then you can do a pan scan and see whether you can pick something up. But it's not the dynamic range, it's not very sensitive. To obtain sensitivity, we have to use a triple quadrupole instrumentation. What colleagues in drug metabolism do for a living can be done here too. So we choose in the first quadrupole. I have to describe that. We have the PEG peptide. Now, they elute all at the same time from the chromatography. The PEG peptide, which is 35 kilodalton. We put a little bit of energy on it, and we get that fragment at 659. And then when we fragment that, we get the ion at 271. If we do the same thing, again, the same PEG peptide, we create the fragment by mild activation. Then our parent ion is at 665, and our daughter ion is at 287. So that's what we're doing here. We put the first quarter ball at these two molecular weights, which then selects these molecular weights. And we, at the same time, put the third quarter ball at these molecular weights. So only molecules which have the proper C-terminal molecular weight and have the proper pKa will give a blue signal. And only molecules which are by 16 higher as a parent fragment and do have the 287 as oxidized pKa would show up in red. And that's what we saw. This is shifted for illustration. It's not the time separation. They are at the same time. So if you look at the GLP lot, that's the response we're getting for the oxidized pKa. And for the GMP lot, it's essentially gone. So we could say something about lot-to-lot consistency during the development phase, stability indicating method for shelf life determination. It turned out that oxidized pKa was really introduced from the very start. I mean, as an activated building block, it was there already. And it turned out not to, once that was removed from this building block, it turned out not to be a problem at all, because we didn't observe any increase of oxidized pKa during the pegylation process, nor did we observe any change in the pKa during stability studies. So we don't have to worry about it, really. But we had to when we didn't know exactly what's going on. 2 minutes. Okay. That's going to be. Aside of mass spectrometry, we use triptych maps to cut the peptides in 3 pieces. So we got 2 real peptides and the third one which is pegylated. So these are actually the 3 peptides. And we did look at the amidation with triptych maps. That's the problem area here. Where we have the amidation here too, but at the C term we have the amidation too. And what we observed is when we looked at our digest that we really had to watch out, because if we did a time course, we couldn't just make a digest, quantitate the amidation. We had to use a time course and then extrapolate to the time 0, because we did produce the amidated species during the digestion, and we did produce artifacts. That's a digestion artifact. Is if we look at the kinetics extrapolated to time 0, there's nothing there. So we had to extrapolate to time 0. The epimerization is another hot spot, we call it. It's not really a problem, but we have to watch out for that. We did observe an epimerization at that asparagine. So it was a deamidation and an epimerization. I don't quite fully understand that epimerization, because normally asparagines, they do the epimerization and then convert into the aspartic acid. In this case, I'm almost certain that it is actually the asparagine with epimerizes. Maybe not 100%, but almost certain. But it maintains the molecular weight. It doesn't convert into an aspartic acid. And in the triptych digest, that looks like this. We had quite a high amount that was produced during the pegylation. The basic environment with the piperidine did pyrimization to that, and that's what was our, during the triptych digest, not changed, so no time course there, no extrapolation to time 0 needed. And then there are 2 deamidated forms also observed, which we extrapolated these values to time 0 to obtain the purity values. I think I'm going to jump through it. Yeah, I want to. Are the slides available later? So the slides are going to be available for us later. Okay. All right. So then, of course. Now, the thing is, of course, again going back not too long, that really, if we are talking about complex drug product, not pegylated. Also, Volker Schellenberger, he promised me that he will make my life easier with the PEG. So he told me, he promised me that he will solve all my problems with having the extent monodisperse and non-polydisperse. I don't want to promise him, but I told him I will find the problem still. So just because the CMC issues. And then there's some acknowledgements. I don't want to go to this list, but I really want to mention Roche Boulder, because they did the project went from GLP. We went into the phase 1, and we were ready to go into phase 2. And our colleagues in Roche Boulder, they did a phenomenous amount of work and the job to scale that up, to improve the quality of the peptide, to change, to tune the pegylation such that we really had a much better product than what we had to go into phase 1. And we had all that material ready. And it was a very sad moment when we actually had to decide that the project will have to be terminated. And the reason why it had to be terminated is because of the known PEG issues with accumulation and all that. So that's not it's never a happy moment, but it was a fun project after all. A lot of experience gained with it. Okay, thank you very much for your attention, and thank you for thank you, Walid, and the organizing committee for having me here and bearing with me.
Yeah. Thank you, Hanspeter, for showing us how difficult it is to characterize pegylated peptide.